Welcome students to the second lab of the digestive system histology. In this lab we are going to talk about the histology of the accessory digestive organs, namely the salivary glands, the liver, gallbladder, and the pancreas. So we start with the salivary glands. Now, the major salivary glands are three: the parotid, submandibular, and sublingual. So we start with the parotid salivary gland. We see that all these are the secretory portions of the parotid salivary glands. You can see that these are serous acini. We talked several times how to differentiate between the serous and mucous glands. The serous glands take up stain, so they are colorful, whereas the mucous glands have no stain, so the cells appear empty or white. So here in the parotid gland, we can see that all the acini are stained because the parotid is formed of serous acini. In addition to the acini, we can see two ducts. This is one duct, and this is another duct. Notice how here we have small cuboidal cells with round nuclei, whereas here we have long columnar cells with oval nuclei. This is one of the main ducts. So this is a parotid salivary gland with serous acini. The submandibular salivary gland. Here mostly we have serous acini. All these are serous acini. In addition, we have some mucous acini. Those that have no color or are white in color, these are mucous acini. But actually, most of these mucous acini are mixed with serous demilunes. So here we have an acinus. This part here, the colorless part, this is the mucus cells, and this crescent-shaped part is the serous demilune. In addition, here we can see several of the ducts of the gland. These are the ducts, paler in color. So if we take a mixed seromucous acinus and we examine them under the microscope. So if we use the rapid freezing method, which is commonly used for electron microscope, we'll get this image. As you can see, we have mucus cells and we have serous cells, one beside the other, one adjacent to the other, both of them found within the acinus. So here, this is the actual feature, this is the actual shape of the acinus, and we do not have a demilune. But if we use the conventional methods or routine methods for preparation, as is done for the light microscope, the mucus cells will swell. The mucus cells will increase in size and they will push the serous cells to the outside, leading to the formation of the serous demilune. So serous demilunes are not actual features of the acinus, but it results from the way we prepare the tissue for study. Finally, we have the sublingual salivary gland. It is formed of mucus acini with serous demilunes. As you can see, everything here is empty looking, so these are all mucus. However, we can see that they have a serous demilune, so this is an acinus. This is the mucus part, and here we have a small crescent shape, and this is the serous part. This is the serous demilunes formed of the serous cells. So the sublingual salivary gland is mucus with serous demilunes. So the parotid is serous, the submandibular is mostly serous with mixed acini, mucus with serous demilunes, and the sublingual is mucus acini with serous demilunes. The histology of the liver. Now, the liver is formed of several polygonal lobules. In this specimen here, the lobules are indicated by dark boundary. Now, of course, this boundary is formed of connective tissue. However, in humans, this boundary is the boundary between the lobules is indistinct. So actually, this specimen is not from humans. So here we have this lobule, and here we have another lobule, and all these are hepatic lobules. So again, although here we can see boundaries, but if we see boundaries, this means that this specimen is not from a human. So this image shows the hepatic lobule. The hepatic lobule, although it is hexagonal in shape, but not necessarily that we can see that clearly in the slide. In the center of the lobule, we have the central vein, and all the thin lines that appear to be, all the thin white lines that appear to be radiating towards the central vein, these are the sinusoids. The lobule, at each corner, we have a portal triad. Here we have, for example, and here, these are all portal triads. Now, it is hexagonal, so there should be 6 corners. So for each lobule, we have 6 triads. However, here we can only see, we do not see all of them, we see only 5 of them. But there's supposed to be 6 for each lobule. In this magnified image of the previous slide, here we have the central vein of the hepatic lobule. All the white spaces, these white spaces, these are all hepatic sinusoids. All these thin white spaces, these are the hepatic sinusoids. On the other hand, the highly acidophilic cells, these are the hepatocytes. These are the hepatocytes. All these acidophilic cells, these are the hepatocytes. Why are they acidophilic? Of course, because they contain numerous mitochondria. Other types of cells may be found in the hepatic lobule, of course, but in the lab we are going to only talk about the hepatocytes. In the portal triad, we have, from its name triad, we have vein, artery, and hepatic duct. All these structures are embedded within loose areolar connective tissue. So a vein, an artery, and a duct embedded in loose areolar connective tissue. So how to differentiate between the vein, artery, and bile duct in the portal triad? Now, regarding the size, the vein has the largest size, and the artery and bile duct are smaller than the vein. The shape of the vein, look at the vein, it is collapsed, whereas the artery and the bile duct are round in shape. The wall, the vein has the thinnest wall of the three. The artery has the thickest wall, and the bile duct has a thick wall, thicker than the vein, but not as thick as the artery. The epithelium, the vein and the artery are lined by simple squamous epithelium. As you can see, here we have the nuclei of the squamous cells, whereas the bile duct is lined by simple cuboidal epithelium. Now this is why the bile duct is thicker than the vein, because it has a simple cuboidal epithelium, whereas the vein is simple squamous. But in the artery, in addition to this epithelium, we also have a thick muscular layer. All these are muscles. This is why it has the thickest wall. The histology of the gallbladder. The gallbladder wall is formed of mucosa and a muscular coat surrounded by adventitious serosa. So we do not have a submucosa here in the gallbladder. The mucosa of the gallbladder is formed of epithelium and underlying lamina propria. Now the mucosa is thrown into folds. As you can see here, we have folding of the mucosa. Of course, these folds are formed of epithelium and the lamina propria. And then beyond that, we have the muscular coat, and the outermost layer could either be a serosa or an adventitia. Remember that the fundus of the gallbladder is outside the liver and covered entirely by peritoneum, and therefore the outermost layer here is serosa, whereas the body, for example, of the gallbladder lies against the liver, and therefore the part of the gallbladder in contact with the liver is not covered by peritoneum, and therefore the outermost layer here is adventitia. Here we can see the epithelium of the gallbladder. Notice here we have long cells with oval nuclei. So these are columnar cells, and the epithelium here is simple columnar epithelium. Finally, the histology of the pancreas. Pancreas is a mixed exocrine endocrine gland. The exocrine portion of the pancreas is formed of serous acini, and these take up stain, and all the basophilic structures. These are all the exocrine portion of the pancreas, the serous acini. The lightly colored circular areas, circular structures seen scattered among the exocrine portion. These are the islets of Langerhans, or the pancreatic islets, and these are the endocrine portion of the pancreas. This is a magnified view of the exocrine portion of the pancreas. Here we have an acinus, and here there's another example. So all these are acini of the pancreas, and they have the typical features of serous acini. We have intense basophilia in the basal part of the cell. So the basal part of the acinus is intensely basophilic. Why? Because in this region here we have the nuclei and the rough endoplasmic reticulum, whereas the apical part of the cells, the apical part of the serous cells, which happens to be the center of the acinus, contain the secretory granules. Secretory granules could be either light basophilic or even acidophilic, as can be seen here in the pancreas. So this is the exocrine acini of the pancreas. And by this we come to the end of the lab of the histology of the digestive system. Thank you and good luck.